No. Anton Dibari. Anton Dibari is the father of plant pathology, also called as modern mycology. Father of modern mycology and plant pathology. Anton Dibari first. Where is the gene? Oh, okay. Anton Dibari first studied in a about the plant diseases. You know which one is the first studied plant disease? Which one is the first fungicide? First, I already told you while studying in the Panjai. In the second unit also we discussed.
Brown spot flies.
Uh,
Brown spot flies.
Brown spot of eye. No. Okay. You know the
Plus.
Phytophthora infestans, late blight of potato. Late blight of potato is caused by Phytophthora infestans. The first fungicide discovered to control the disease. It is not discovery, it is spontaneous discovery. I already told you. Whenever the president or secretary who they are coming, usually what we do, lime and bleaching powder we pour the both the side of the road, corners of the road. Yes,
Yes, sir.
that mixture cured disease of the potato, late blight of potato. They are not sprayed any fungicide pesticide, but they try 2-3 years. At the time of 1860, 1867, they faced lot of problems because of no potato crops in Ireland. European people, they eat potato as like how we eat the rice. They totally lost the potato. Then they tried to control the disease, but they couldn't. But spontaneously, naturally, it cured by our normal condition, normal habitat, which we pour the bleaching powder and lime. That is the first fungicide, that is the Bodax mixture. Okay, we will go for our diseases. First fungicide is the Bodax mixture, that is the mixture of lime, bleaching powder, and water. This is the first fungicide, the Bodax mixture. Okay, so many famines, starvations happen all over the world because of the small microorganism, either fungi or bacteria or virus. Ireland famine is the first famine identified. And the French famine, French prawns. In the prawns also, famine happened because of the small fungi that caused lot of damage to the grape. In the prawns, grape crop is a very wide growing crop. Grape is the very wide growing crop in the prawn. Okay. That wide growing crop affected with the Plasmospora viticola, that is the fungi that cause downy
Disease.
mildew, downy mildew of grapes.
Downy mildew. Okay.
Yes, downy mildew of grapes that caused by Plasmospora viticola that damage all the field of grapes. That so many pathogens which cause lot of damage, total germination of the all the crops. You know, Bengal famine, that Bengal famine is caused by, Pericularia varize, gibberella, the Pericularia varize, magna forte, Pericularia varize. That Pericularia varize or magna forte that cause damage to the paddy crop. You know, in the Bengal, West Bengal and West Bengal, Bangladesh and West Bengal, total rice crop are damaged at the time of 1943 to 47, before independence. Before independence, Bengal people are get starved because of no rice crop. Total rice crop are damaged with a small fungi that is Pericularia variety. You know, here we are going to study that also. You see the blast of rice. That disease caused lot of damage to the cause death of the so many Bengal people. Okay, and then the bunchy type of banana in the Kerala. All the banana grow very high lot of area in the Kerala. You know the banana Kerala, Tamil Nadu and Kerala banana growers grow very wide, large area. That Kerala bananas also get damaged with a small fungi. You know the Sri Lanka famine, that has caused the coffee rust disease that caused by small fungi that damaged all the coffee crop of the Sri Lanka. Now Sri Lanka are growing tea crop, tea plants instead of coffee. Total coffee crop are damaged. They altered with the tea. So all the world economy and specific country economy being affected with the small fungi, very small, minute fungi which we couldn't see with our naked eye also. That caused a lot of damage, starvation and death. Okay, that is a brief introduction about the plant pathology. Okay, these are the some famines: Ireland famine, French famine, Bengal famine, Sri Lanka famine, and Kerala banana, bunchy type of banana. Okay, these all are cause lot of damage to the crop. Okay, here we are going to study the first one. What it is? First we are going to study the rice diseases. Not all the rice diseases, only two rice diseases we are going to discuss.
Okay.
There is almost 1, 2, 3, 4, 5, 12 type of the major diseases are there that cause damage to the rice. 12 major, you see here in the image. Could you able to see this?
Yes, sir.
Yeah, these 12 major type of the diseases which cause the damage to the rice. But we are going to study only two. These all the diseases which we are going to discuss are very, very important in examination point of view. Of these 1, 2, 3, 4, 5, 6, 7, 8 diseases, 2 essay question will come. Okay. So you have the
Sir,
chance to get
all 12 diseases are important, sir.
all 8 of the 8 diseases. 4 diseases will come. You have to write the 2 diseases.
Okay, sir.
And 2 diseases,
Mm.
each one having the 10 marks,
Okay, yes, sir.
Or otherwise it asks the short answer, life cycle of any disease or control measure of any disease as a short
Okay,
answer. Suppose control wheat rust, control measure of loose mud of sorghum, or control measure of sugarcane, or control measure of
Tick.
or groundnut or life
Okay.
cycle of ticka disease.
Mm.
Like that, they are the question. Okay,
Okay, sir.
short answer and essay question. Two short answer will come, one or two essay question will come.
Okay.
From this,
Okay.
that means majorly 20 to 30 marks
Okay.
it covers. Okay,
Okay, sir.
yeah. Then we will go each and every disease. You see, Buccaneer disease. First one, rice disease. We are going to discuss two rice diseases. One is the Buccaneer disease. Second one is the blast of rice. Second one is the blast
Blast
rice.
of rice.
Okay. These two diseases we are going to discuss. Okay.
Okay, sir.
Okay.
First one is Buccaneer disease. You know, bakra. Bakra antama? Bakra అని ఎవరు అంటావు?
బక్రా అయిపోయిండు ఎవరు
గోడ్ని అంటావా?
Cool.
మనుషులు కూడా
Sir.
అంటాం బకరా.
అరే బక్రా అయిపోయిండు అంటే మనం చెప్పింది
నమ్మేవారు.
బక్రా
తెలివి తక్కువ వాడిని మనం బక్రా అంటాం. అవునా? కొంచెం మనం చెప్పింది చేయలేకపోతే, చెప్పినా వినలేకపోతే, ఏ పని చేయలేకపోతే
బక్రా అయిపోయిందంట.
సేమ్ ద ప్లాంట్ విచ్ ఆర్ వెరీ సిక్ అండ్ మ్యాడ్ లైక్ దట్స్ వై వి నేమ్డ్ ఇట్ యాజ్ బకనే డిసీజ్. తెలివి తక్కువ మొక్క.
ఓకే.
Total plant will become sick and fall down to the earth, and all the roots are damaged. Major first it causes the disease to the roots. Okay, from the root, from the soil, it attacks the roots. All the roots will be damaged, decomposed, degrade. So there is no tip, there is no support, there is no strength and support to stand upon the soil. So it fell down. It fell down on the soil. Okay. That disease is caused by
Buccaneer disease.
Buccaneer disease that caused by
Fungus.
జిపరెల్లియా ఫిజికోరియా
Fusaria manslifer. Fusaria manslifer.
Gibberella fujicorae. This we already told you this in the phytohormones. Whenever we discuss the phytohormones.
ఓకే.
Gibberellins.
Gibberellin. That gibberellin extracted from this Buccaneer diseased plant. That gibberellin extracted from the Buccaneer diseased rice plant. That's why we named it as gibberellin.
ఓకే.
Gibberellin, that is the phytohormone. You know that cause for the ripening. That means more fruiting, wide growing of the stems. You know the cauliflower, cabbage, wide width growth of the cauliflower and
Growth
cabbage.
promoter.
Rosette flowering. For all that we use this gibberellin. That gibberellin is extracted from this Gibberella fujicorae fungi, which cause the Buccaneer disease on rice.
ఓకే.
Buccaneer disease on rice. Even though it is a disease, it causes effect on the rice. It is also using as a phytohormone. Whatever the proteins which are released after the disease caused to the rice, that proteins extracted, that extracted proteins are gibberellin. That alkali, that is gibberellin. That gibberellin we use as a phytohormone. You know, auxin, gibberellin, cytokinin, abscisic acid, ethylene.
ethylene.
That gibberellin is extracted from the Gibberella fujicorae.
Yes, we studied
that called
Buccaneer disease.
Buccaneer disease in the rice. Okay, and you know abscisic acid release extracted from the cotton. Abscisic acid, that is stress hormone. That means that cause dormant.
Seed dormancy.
Yes, seed dormancy. Encourage the seed dormancy.
Abscisic acid and ethylene.
Yes.
The two are the growth promoters. 3 oxygen, gibberellin, cytokinin. Abscisic acid and ethylene are growth inhibitors.
Yes, inhibitors.
Okay.
Yes.
Yeah, we are studying the Buchaney disease. What the symptoms? First, the symptoms which appear on the leaf, spot leaves are there, spotted brown spot leaves. Whenever it is primarily attacked, and then it goes toward to the phloem tissue, it goes to the roots, and all the roots will be damaged. The root will be decomposed and damaged. The cause for the wilt totally fell down upon the soil. Okay, that's why it looks like a very that means delicate. Okay? Very delicate. It caused the all fell down, all death of the plant. Okay, that is Buchaney disease. What it is?
Buchaney disease.
Buchaney disease. You can see the gibberella musicori fungi. You know the symptoms we already discussed. How we identify that, and then what is control measures? How we can control? What the control measure? Every disease, whatever the disease, what you have to write? Disease character and disease called pathogen. What the pathogen? How the pathogen? And control measure. These 3 steps must in all the diseases. Whatever the disease, whenever you are going to write in the exam, these 3 points are very, very important.
Yes.
Okay. Disease characters, how it is, where it starts, when it starts, what the first symptom, later symptoms, or how the plant, whenever it attacked with the specific pathogen. And the second one is the pathogen, how the pathogen characteristic, whether it is a filamentous or sporous, how it is, where it goes, when it attacks, that pathogen character, how the mycelium, how the spores, how the fruiting body, how which part of the life cycle it affects on the host. That is pathogenic character. And then next, control, how we can control. Majorly, we can control by 3 methods: cultural method, biological method, chemical method. Biological, cultural, and chemical method. Biological method means
Crop rotation.
Sheets. See.
biologically by using other biological.
Like a crop rotation also.
And what?
Grow predators by growing predators.
Study the history of
Yes.
Life cycle.
Seed.
By observing the life cycle.
Yes.
By observing the
and introduce
life cycle.
predators.
Yes, you can see.
Mixed crops. No.
What are those? What are the cultural methods?
Seeds. Good. Use good
Removing of the
seeds.
weeds, whatever the secondary host or any other parasitic host are there, we have to remove it. We have to maintain the proper healthy condition. Field area, whatever the field area where we grow, that area should be neat. There is no weed crop surrounding of it.
There is no moisture.
Proper
Moisture.
Dry area.
sanitation, sir.
Yeah, usually in the paddy crop, moisture is always needed. In the paddy, all the rice crop. Okay, that all. And the biological, what are the biological? Cultural.
Change
Where
the crop. Mixed crops.
You see,
Should
how the
avoid
biological
affected
control.
species.
By using the other biological, the fungicides and pesticides, some of, biological fungicide, biological control. trichoderma, bacillus, Fusarium, that we are using as a biological pesticide. Usually we spray the sprinkles of bavustin, monocotopas, that chemical pesticide. That chemical pesticide causing the lot of damage to the farmer.
Pollution.
Also, that is being inside the seeds that we are taking inside, that causing for the cancer, other diseases, lung diseases, heart diseases, that all causing, and also it causing environmental pollution. Instead of that, we can use the biological control. Fusarium, trichoderma, bacillus. Fusarium, trichoderma, these two are fungi. It kills the other fungi and bacteria,
Yes, sir.
like penicillin.
Yes.
Yeah, these biological control
Enemy
method.
animals, like predators, enemy animals.
Yes,
Introduce.
yes. And also, like that, chemical control is necessary. Majority of the farmers are using chemical control. The insecticide, fungicide, bavustin, monocotopas, carbendazim, and these are we use to control. Okay. Instead of all this, one of the best method is we have to show the resistant variety. This is resistant variety.
Hybridization, sir.
Which seeds is having the resistance to that disease? That varieties we have to show that, so that we can make it free from this disease. That is the best method. Best method is disease resistant variety. Second best method is biological control. And the third one is the cultural
Cultural
maintenance.
farm.
And then last to least one is chemical pesticide, chemical treatment that cause lot of damage, but there is a need to control the disease, to stop the widespreading of the disease. Okay.
Okay,
That
okay,
are
sir.
the
Okay.
control measures of the buccaneer disease. This is the first disease of rice. Okay.
Okay.
And then the second disease, blast. Where it is?
Wheat
Where it is?
blast.
Where it is? Blast. Oh, yeah. The blast disease, which caused major damage. I already told you, Bengal famine. I told you now, Bengal famine.
Mm.
That Bengal famine is caused by
Blast.
because of blast of rice. Because of blast of rice. Okay. You know, sorry, where it is?
Screen not visible, sir.
Did not, still not visible.
Sir, this is
Visible, sir,
blast.
but no.
Visible, but
Oh, okay, then. Where it is? What happened to this? Okay then. Okay, now it is. Okay.
Yes, sir. Yes, sir.
Yes, sir.
Yeah.
Yes, sir.
This is the blast disease. The first, whenever it attacks on the leaf you see how it is. All the leaf will be turned into brown, yellowish. Blast means kalipaina machal. The blast. కాలిపోయిన మచ్చల్లాగా ఉంటుంది. లైక్ ఎండుగడ్డి అంటామా, వరిగడ్డి ఎండుగడ్డి అంటామా.
Yes.
Yes, sir.
After harvesting it will dry na. The dried one we call it as ఎండుగడ్డి, వరిగడ్డి అంటాం, అవునా.
అగ్గి తెగులా సార్?
Ah,
అగ్గి తెగులు
yes, అగ్గి తెగులు, ఎస్. లాస్ట్ మండిపోయినట్టుగా, మండితే ఎట్లయితే కాలిన మచ్చలాగా అవుతుంటుందో, like that it all totally brownish and dead necrotic spots will appear. And all the whatever the pedicle, that means spike, కంకి, రైస్ కంకి ఉంటుంది కదా, పానికి spike, that spike at the base of the spike totally damaged. So the తాలు అంటామా, there is no grain, there is no grains inside the husk, only husk will be remain.
లేయర్ ఉంటుంది, లోపల రైస్ ఉండదు.
outer layer husk only will be remain, there is no grain inside.
Yes, sir.
That caused by
Blast.
by which fungi it is. I already told you, Pericularia variae, Magna forte, Bengal pemine. Told you na?
Yes.
Bengal pemine, Pericularia variae. That disease is caused by Pericularia variae. The later name as sexual stage is Magna forte. Magna forte variae. Magna forte variae. After the sexual stage confirmation, they changed it, named as Magna forte. Okay, you can see the spores, conidial spores, how it is, the fungi, pathogen. First disease symptoms are, what are the disease symptoms?
Necrosis.
That means blast spot, that means కాలిపోయినటువంటి మొత్తం. And fire అయితే ఎలా ఉంటుందో, brownish spot, and it will become totally turn into dry. All the leaf will be dried, as like a dried leaf, and the spikelets, all the spikelets are damaged. The spikelet at the base, it will be decomposed and totally occupied by the fungal space. The spike occupied by the fungal space, so the spike will be bent. Instead of straight spike and with the number of seeds, number of fruits, that all the spike will be turned back. You see here, turned back. You see it turned back. With the total, all the leaf will be dried as like a grass, dry grass. Totally damaged. First spot will be look like this, and then it turn into totally brownish, and the spike will be bent back, and in the spike there is no fruit. Only husk, outer layer only will be present. That is the disease symptom. Okay, and then next what we have to study, the pathogen. How the pathogen? Pathogen is Pericularia variae or Magna forte variae. Okay, how it is? It having the conidia and conidia 4. You see the conidia. Conidia 4, there are a bunch of the conidia are there. You see the bunch of the conidia. These conidia form. Conidia will be widespread and it enter into the leaf through the stomata. Leaf through the stomata, and it enter inside, and it totally whatever the living tissues, chloroplast tissue, all will be taken out and eaten by these fungal spores. So all the greenish leaf will be turned into brown and black, dark black or brown patches. Okay, that's why we calling it paddy blast. Paddy blast. Okay, and how we can control? 3 types. You know the control measure? Cultural,
Cultural,
Biocontrol,
Bio.
chemical method,
Pet.
Biological, cultural, and chemical. Okay, where we are, we have to close up. Biological, cultural,
Chemical.
yeah, chemical. Oh, where it is? Okay, what are those? Biological. You know the biological means we already told you can write the same type of the biological and cultural to all the diseases, but chemical is different. Okay,
Okay.
majorly chemical is different. Cultural method, you know what are those cultural methods?
Crop rotation.
Crop.
Crop rotation. And
Mixed
what are the
crops.
biological one?
sanitation,
Fish crop.
What are the biological?
Use high quality disease species.
And
Hybrid seeds.
biological means already I told you now. Trichoderma,
ba-
Fusarium, Bacillus,
bacillus,
Bacillus.
fungicide,
penicillium,
biological control method. Instead of chemical,
Okay.
using
Okay.
of the biological fungi to kill the fungi. We are using the fungi to kill the fungi as a predator. Then we can control the disease. Eco-friendly. There is no damage to any field or farmer or any other. Okay. Cultural means we have to maintain the need. We have to maintain that means hygienic condition, and we have to remove all whatever the weed crops nearby or surrounding or in the crop. We have to make the
sanitation,
distance. We have to make the distance between the two rows of the crop for the aeration and for the removal of the whatever the other fungicide, fungus or bacteria, viral
Pest.
spores, uh, and also to
disease-free seeds,
uh,
Fertilizers.
uh, and
disease-free seeds,
usage of the optimum level of fertilizer, NPK, nitrogen, potassium, and phosphorus. If the
Yes.
more potassium, if we use the more potassium, then will be automatically fruiting will be more. It will come very early. Fruiting will come, so that it may be some resistance before the disease happening. The all the field will be fruiting. That means ripening condition. In the ripening condition, there is a need of optimum usage of the fertilizers, NPK, urea, DAP, phosphorus, and 315, 320Y are there now. That usage, apply of the nitrogen and phosphorus. Only 25% below, not more than that. మన వాళ్ళు ఏం చేస్తున్నారు, యూరియా కట్టల పొలాలలో జల్లేస్తున్నారు ఎక్కువ తొందర పెరగాలని. అవునా? డోంట్ యూజ్ లైక్ దట్. ఆప్టిమమ్ లెవెల్ పర్ ఎకర్ how much? Only 50 kgs only. To 25%, that means not more than that. If you use more than that, it grow very high with a delicate, so that there is no chance to give the yielding. Okay. Chemical pesticide, you know the chemical pesticide, especially copper sulfate. You know, whatever the fungicide, we can control by the copper sulfate. You know, even lime, calcium. With the lime also we can control. Sunnamantama, lime. With the lime also we
yes,
can
sir.
control the fungal disease. I already told you, first fungicide with the lime and bleaching powder. With the lime, with the copper sulfate, and you see what are those? And you know, by the fumigation also, majorly carbendazim, monocotopaz, and copper sulfate. Okay, and what is this? Azo? You have to read out. What is it? Azoxy? Where you are? Are you looking at the screen?
Azoxys screen is not there, sir.
Azoxystrobin.
Mm.
That is the best chemical that controls. And the second best is the copper sulfate. And the least one is the remaining carbondism, monocotopos, metamorphodine. These all are the major chemical control measures. These are the two rice diseases only we are studying. First one is the Buchaney disease. Second one is the
Blast
of
of
paddy,
us.
blast of rice. Okay. These two
Okay.
are very, very important. From Buchaney disease, disease plant, we are extracting gibberellin phytohormone. Blast disease also, it caused famine, Bengal famine, that caused the starvation of the Bengal people 2 years. Okay, these two are very major, important GK, general knowledge point of view, general science point of view, even in your examination point of view, the very, very important. As a botanist, you should know where phytohormone that is extracting from the diseased plant. Tell me the komaka. Buchaney diseased plant. Okay, Gibberella fungi. And you know, Bengal famine caused by Pericularia variety. At the 1943, 43, 44, 43 to 45. Bengal famine. What? What happened? What?
West Bengal rice crop damaged.
Total West Bengal 1943 to 45, 2 years. So many people have died. You search in the Google Bengal famine. So many dead, so many are sick, so many are very delicate, sick, so many they don't have any food to eat, so many are migrated to other places, the 43 to 45, because of this disease. Okay, these two are very important diseases. Okay, then where we are? The next is sorghum. Before sorghum, we will study the wheat. You see, wheat. Wheat also having the almost 16 major diseases. But we are studying only one or two. How many we are going to study in the wheat?
Wait, only one.
Wheat rust. And also loose. What it is? Second one is
Loose mud. Loose mud.
loose matter. Yes,
Mud.
Vistillado. It's called by
Yes,
Vistillado.
sir.
Yes, yes. Yes. Two diseases. Okay. One is the first one is the
Wheat blast.
Wheat rust.
Mud.
Rust.
to see the various diseases.
Black rust. Rust.
Black or stem
Black
rust of
rust,
it.
sir.
Black stem rust. You know,
Loose mud.
rust of
Next one.
wheat. Wheat rust. Okay. You see the wheat rust disease. It is stem rust of wheat or wheat rust disease is caused by Puccinia graminis triticale. Puccinia graminis triticale fungi. Puccinia fungi. Which fungi it is? Puccinia. You know the Puccinia life cycle. They ask the question as a Puccinia life cycle also. Even Puccinia life cycle also you have to write this disease. Same. Stem rust disease. Okay. And you know the macrocyclic life cycle. It is the macrocyclic life cycle. That means the life cycle is very lengthy. Very lengthy life cycle. It is not life cycle is not ending with only one type of the spores. It releases the 5 type of the spores. How many?
5.
5 type of the spores.
5 types.
Yes, it releases the 5 type of the spores. 5 different type of the spores. It having a lengthy life cycle. That means one type of the spore, it germinate and it mature and form that another type of the spore. Another type of the spores which fell down on soil and it germinate Basidiospores. And basidiospores which attack on other host and it cause the disease to the other host. And this called pycniospores and ecdiospores. 5 type of the spores. What are those? Pycniospores, ecdiospores, basidiospores, uridospores, ptylidospores. What are those? 5 type of the spores. How many? The first one is
Five.
ecdiospore. Second one is pycniospore. Third one is basidiospore. Fourth one is uridospore. Sixth one is ptylidospore. Fifth one is ptylidospore. These 5 type of the spores which release. That's why this life cycle is very lengthy. It took long time. It depend on 2 hosts. It depend on
Two hosts.
2 hosts.
Two host.
Yeah, where on which plant it having the sexual life cycle, that we call as primary host. Primary host, first host. Primary host is here wheat crop. Primary host is
Wheat crops.
wheat, godumalu, wheat. Okay, not weed. Weed means kalpmokka. Wheat, goduma. Okay. Wheat crop is the primary host. That means its sexual cycle is happening in the wheat. Okay. That's why it's the primary host. And the secondary host is weed, kalupu mokka. Barbary plant. You know, barbary plant. Not barber. Huh? Barber kaadu. Barbary plant. Barbary, it is a weed widely growing in the nearby crops on barren lands, wastelands. That is the weed plant. This is the weed plant, Barbary plant. This is the secondary host. Primary host is wheat. Secondary host is Barbary plant. Okay. It caused major damage to the wheat crop. Major disease in the wheat. One of the major disease in the wheat is one of the major disease in the wheat is
Trust. Wheat rust.
wheat rust.
Mm.
One of the major disease that is wheat rust. Okay, you know it is causing by
Pasinilia graminis. Pasinilia graminis.
Graminis. Vaccinia is fungus name. It causing the disease to the Graminae family, all the grasses family. It causes diseases to the grasses family. Not only that, to the wheat, jowar, bajra, sorghum, grasses, Graminae family, Poaceae family, and triticide. You know, especially it is causing disease on Triticum vulgare. Goduma scientific name. Wheat scientific name is
Triticum vulgare.
vulgare. That's why we call it as Vaccinia graminis triticide. It is affecting on the triticum plant. That's why triticide. Vaccinia graminis triticide. Okay. It having the lengthy life cycle. That's why life cycle we call as macrocyclic life cycle. Macrocyclic life cycle. Okay. And
5 types of the spore.
5 different type of the spores. What are those? Ichthyospore, pycniospore, basidiospore, uridospore, ptylidospore. Are you noting down these points or only
Yes.
listening?
No.
Noted.
Please note, otherwise you can't remember again. We are not Swami Vivekananda to remember at once. Okay, we are normal people. Okay,
Okay.
then we need to note down. Yeah, here it having the 2 hosts. Primary host is the wheat.
Primary, secondary.
Secondary host is the Barbary plant. That is the weed plant, widely grown weed, kalupu mokka. Okay, That's why, as a cultural method, we discussed earlier, cultural method. We have to remove the weed crop nearby or within the crop so that we can control the wide spreading of the disease.
This is
First major, the biological, cultural controlling method, removal of the weeds, widening in between the plants, pre-aeration, pre-sunlight should be to the all the crop. Here, if you remove the barberry, here it is. If you remove the barberry plant, automatically this life cycle will be stopped, so that we can control the Puccinia fungi, Puccinia, so that it will not again attack the wheat crop. So remaining wheat crop will be safe. Yes. Yeah. Here you can see the first whatever the esiospore that released from the weed plant. You see esiospores here, the plastic shape. Here you see the bell shape rooting body. Bell shape. From there, the esiospores release. These esiospores attack the wheat. No, it will not attack directly wheat. It will attack the barberry plant. Okay. The first, whatever the esiospores, whenever it attacks on the wheat, they get convert into uridospores. What it is? The first spore which attacks on the wheat is uridospore.
spores.
Urido means red color spores, which are in red color. That's why we named it as uridospore. Which are in red color, which are in the conidiophore and conidia. Conidiophore and conidia. Conidia, which are in the two nuclei. Conidial
nucleus.
layer
Yes.
is in the red color. It having the two binuclei. Binuclei. Yeah, it usually it starts germination in the winter season. It is uridospores will germinate. That whenever it is uridospores attack on the leaf, it enter into the leaf through the stomata, and it widespread and it forming the saucer shaped fruiting body. There, the number of spores, iridospores, will be formed with the conidia and conidiophore. Number of iridospores will be formed. Okay, that are in red color. They having the resistant. You know, these are also called as resistant spores, which are in the eye resistance. These are also called as resistant spores.
Resistance.
And also called as repeated spores. Repeatedly it causes the disease on the wheat. If suppose one plant causes the disease, rust disease. Rust means rust. Kunkuma tegulu antam. Kunkuma tegulu, rust.
Red color.
Red color powdery spots untai. Patches untai. Red color powdery patches on the leaf. That's why rust. If the black color powder is, that we call as smut, katika. If it is a black color powder spot, that is smut. Bajra. In the bajra, smut disease will come. In the wheat, rust disease. Okay? Rust and smut. What happened? Rust disease. Okay. Rust spot. Red powdery spots on the leaf. That's why we calling it as rust disease. First it attacks the leaf and then it attacks also continues to the stem. Stem also will be turn reddish spots, patches. That's why we also call it as stem rust. also called as stem rust. Okay, and then here the iridospores which having the resistance, which are till the autumn season. That's why we also call as autumn spores. Autumn spores. Autumn season తెలుసు కదా, autumn, winter, rainy season, spring season,
Yes.
autumn
Yes,
season,
sir.
autumn spores, not autumn bombs, autumn spores. Okay, autumn spores. This will repeat the disease on the same wheat crop. That's why we also call as repeated spores. Then whenever these iridospores mature and it divides, each spore is having the two nuclei, na? This it take the division so that it forms the two cellular structure. Two cells. Here only one cell, two nuclei in the iridospore. Here in the teleospore, single cellular, single nucleus, double cellular, two cells in it. One, two, two cellular structure. When it takes the division into two spores, at that time, the spores will turn into black. The spores will turn into black color. That's why we calling that. That's why we calling that teletospores.
Black
Teleto,
rust.
telekam, teletospores, katuka laga, black color. It turn into black color. That's why we calling that teleospores or teletospores. Teleospores. Okay.
Pores.
Teleospores. When the teleospores are formed, it get turned into red color patches, turn into black color. Okay. That's why we also call as black rust. Okay. And then these teleospores will not attack, could not attack on the same wheat plant. Teletospores will not cause the disease again on wheat. Okay. Teletospores which release out, that will germinate whenever it fell on the soil, that germinate and it form the basidiospore. You see the basidiospore. Whenever the teleospores which are germinated and it forming the basidiospore, basidiospore, spore. Okay, you know basidiospore, two types we have studied: Pragmobasidium and Teleobasidium. Pragmobasidium means fragmentation, branches, septa present. Each septa having the one spore. But whereas teleospore at the end, the typical only having the spores. That's why you see the basidiospore will form from the teleospore. Okay, that is there in the soil, not on the wheat, not on the weed plant. Okay, then this basidiospore will attack on the I will remove it all, then it will be. Okay. This basidiospores will attack on the barberry plant, weed plant, which are adjacent to the wheat. Barberry plants are also there, attached adjacent nearby to the wheat plant. The spores will attack on the barberry upon the leaves. Okay, the spores will form the pycnidium. You see the pycnidium, plastic, perithecium, pycnidium we have studied yesterday. That simple osteoporous, plastic, fruiting body. That form here in the inside, it having the spores, that pycniospores, that again that attack on the lower side of the leaf. Again, lower side of the leaf, and lower side it enter inside the leaf, and it formed You see, palisade structure, and there from on each the spores will be formed. These four chain-like structures will be formed that are ichthyospores. What are those? Ichthyospores. Isidium. Isidium, bell-shaped rooting body is called as isidium. We have studied in the lichen. Isidium, Soridium, Isidium. చదివాం కదా నిన్ననే yesterday only we have studied. You forgot. Isidium, Soridium, Cephalodium. We have studied. Are you there? How many are there?
Yes, sir. We are
Yes, sir.
listening.
Isidium. Yesterday we have studied. Pycnidium also yesterday we have studied.
Yes, sir.
Sir.
So again attack the lower side of this Barbary plant and it forms
నేను
a
ఒక టైం వస్తాను.
bell-shaped rooting body. That bell-shaped rooting body release the chain-like spores, ichthyospores. Again, that ichthyospores again attack on the leaf. It causes the disease. Suppose we break the cycle, life
నేను
cycle.
కూర్చోనండి.
If you break the life cycle, that means we remove the
అవునవును.
Barbary plant. There is no chance to germinate the basilospores on the wheat. On the wheat plant, basilospores will not germinate. So there is no repeat of the disease. There is no wide spreading of the disease. So that we can control by removing the wheat. Okay.
Shweta ma'am, please mute.
That is the disease symptom. Here are the 5 spores. 2 spores on the wheat. What are the 2 spores? Viridospore, red color, black color, and 1 on soil, basidiospore, and 2 on barbary, pycniospores and ichthyospores. 5 spores, 2 hosts, lengthy life cycle. Okay, you know the K.C. Mehta. K.C. Mehta studied very wide, broadly on this wheat rust disease. He finally concluded one of the best method, cultural method, he suggested. From that onwards, we stopped South India, stopped the cultivation of wheat. Only North Indians are cultivating the wheat. In the South, only few, here and there, only few people are cultivating the wheat. You know the whatever the spores, iridospores, which are forming at the autumn, mature, that spores again is going to the northern places by the monsoons. Northeast monsoon, southwest monsoon. You know the monsoon, Rutu Pavanalu. Monsoons. So the monsoon wind, the spores are spreading from north to south and south to north. Understand? At the
Yes,
winter season, these are coming from north to south. And at the autumn season, these spores are going from south to north. So that repeatedly spores are repeating 2 times every year. From there to here, here to there to here. So we could not control the disease, rust disease. That's why what the Mehta said, we need to stop the cultivation of wheat in any one area. So southern usually cultivate very less wheat. That's why the stop the cultivation of wheat in the southern states, our AP, Telangana, Tamil Nadu, Kerala, and wheat cultivating in more in the UP, Bengal, and all the remaining northern states, so that we could control the wheat rust disease by cultural practice, cultural control method. Okay, and so many rust disease resistant varieties are there. Okay, and that is the disease resistant varieties and cultural by stopping the cultivation of wheat in one area to stop, to break the life cycle. Okay. And then this is the one of the major disease compulsory question. Major disease and compulsory question. You see here, the iridospores are repeating. That's why we call it as repeating spore. You see, the
Repeating.
iridospores are two cells, each one having the one nucleus. Whereas the iridospores are single cellular, two nuclei. You see, tiliospores are germinating and it forms the basidiospores. Thus, basidiospores will not attack the same wheat plant. It will germinate on the soil and it attacks the alternate host, secondary host, Burberry plant, secondary host, alternate host. There it germinates and it forms the pycnidiospores and ichthyospores. Again, that ichthyospores will attack the wheat. Okay. This is the life cycle and control measure. Major question, almost 90% compulsory question. Okay. The wheat rust.
Yes.
Okay. The pie, so pie type,
Okay.
we have to write about the pie type of the spore, life cycle. The Puccinia gramineous criticide, it having the two hosts, primary host is wheat, secondary host is Burberry, and it releases the pie type of the spore, ichthyospore, pycniospore, basidiospore, iridospore, teliospore. And it having the iridospore, teliospores on wheat, and pycniospores, ichthyospores on alternate host Burberry. Okay, and that all you have to write. The symptoms, how it is turned red to black. That all that you have to write. Okay.
Yes. Okay.
And then the next disease is loose smut. Yes, loose smut of wheat. Loose smut. You know that is caused by Ustilago. Which fungi it is? Which fungi?
Lustilago.
Ustilago. Yes. Ustilago. That Ustilago fungi which cause disease on
Loose stem on wheat bread.
loose smut. You know, smut means kartika,
Must.
black color. Smut, rust means red color, kunkuma.
Yes.
Okay. Black means smut, kartika. Kunkuma kartika. Kunkuma tegulu,
Black
kartika tegulu.
powder. Black powder substance.
No, not black fungus. Black fungus is the
Black,
rhizopus, mucus.
black
Okay.
powder. Sir.
Black powder, kartika.
Yes.
Red powder, kunkuma. Two diseases, kunkuma tegulu, kartika tegulu on goduma, wheat. Okay.
Okay, sir.
You can see here the Ustilago fungi, that is Ustilago sesatum, that cause disease on wheat. You see the whatever the seeds, kernel, that all the seeds which covered with the Ustilago fungi, then you see the spike, skunky, spike, wheat spike. That wheat spike totally, instead of greenish, it totally turn into
Black.
black.
Black color.
Yes, turn into black color. Black powdery structure will occupy all the seeds. It covers black powder. It covers the seeds. Kartika, smut. That's why we calling that smut disease. You see all the kernel, that means seed, occupied by the black powder. That all the black powder is the spores. spores of fungi, Euphyllogo magister fungal spores. The spores occupy the seed, all the spike, conky, all the spike get turned into blackish color.
Black color.
That is the disease symptom. First it attack on the leaf. On the leaf, black spots will occur as like a red spot, red color powdery spot on wheat, like that. Same black color powdery spot. You see the leaf here and there. You see the leaves on leaves. Black color spots are there.
Right.
The black color spot. First it attack on the leaf, then it enter into the xylem and phloem. So through that it goes to the spike and it enter into the seed and it over occupy. Mycelium will be to fully cover. The mycelium cover the seed. So all the spike will turn into black. Okay, that is the loose smut. That is the loose smut disease. Okay. You see, earlier we have studied already these, no? Esiospore, pycnospore, on the leaf. That we have studied. Where do to remove this all the things? These all are occupied. No. Okay, then it is clear. Okay, this also done. Okay. You see, already it is over.
Yes.
And then we are studying the already loose smut of the disease that causing by
Plants.
what it is Eustylago serratum
Right. So
Use
or
Tilago
segmentum.
Sever.
Serratum or segmentum. Serrata means it look like a sa rampam antama. Rampam sa sushina sa gadu sa rampam gostara rampam sa
Yes, sir. Yes, sir.
okay.
Sir.
serrate రంపపు పళ్ళలాగా ఉంటుంటుంది serrate. That's why serrate. రంపపు పళ్ళు ఇట్లా ఉంటాయి కదా,
Yes, sir.
సాక్ ఇట్లా ఉంటాయి కదా. ఎగ్జాబ్లేడ్
Zigzag model.
అర్థం కాదేమో, ఎగ్జాబ్లేడ్ అంటే అర్థమవుతుంది. కర్రలు కోస్తారా రంపంతో. ది serrate.
Yes, sir. Yes, sir. Yes, sir.
That's why Eustylago serratum. And also we call as segmentum. That means segment. It having the sepals, more segments. Chamber. Eustylago segmentum. Eustylago serratum. Okay, it causing the loose smut disease. Okay, and then what are those? What are those control measures? Control measures. Are you there?
Yes, sir.
Yes. Biological
Yes, sir.
control method, cultural methods, chemical methods.
Biological control always, biopesticide, using of the
Yes.
biopesticide,
Mm.
right? Resistant varieties.
Mm.
That is biological control for all. For all, these two only will come. Okay.
Yes,
Yes,
sir.
sir.
And then the next is
Fungal diseases.
where it is.
Grains.
Okay. Yeah. You see, one of the best method is seed treatment. Seed treatment before sowing wheat or even rice also. Before sowing, if we add with the carbondism or sunnap neelu, copper sulfate or calcium carbonate, so that whatever the fungal spores which are there attached to the seed, that all will die. That is the best method, is the seed treatment. Or otherwise, what we have to do is we have to pour the seeds in the light heat water, not the fully boiled water. Light heat water. So that whatever the spores, fungal spores, that will die. Understood? This is the best method, cultural method. Cultural method.
Yes,
These are.
sir.
Yes, sir.
Yes, sir.
Yes, sir.
Before sowing seed, we have to pour the seeds in the light heat water, or we have to mix, cover with the calcium, sunnam, lime, or
Lime
copper
water.
sulfate, or carbondism, or gypsum, unhydrated calcium carbonate, gypsum on top. You know that we use as a plaster of Paris.
Yes, yes, sir.
That plaster of Paris unhydrated. That means no water in that. Fully dehydrated lime is the plaster of Paris. That also we can use as a seed treatment, in the seed treatment. That are the cultural methods. Okay. And then chemical method. What are those chemical methods? Drying of the seeds, or also adding the chemical carbondism to the seeds before sowing. And the wet treatment, I already told you now, light heat water. We have to pour the seeds on the light heat water and we have to take out. Then whatever the spores are there upper side of the seed, that all will die. Okay. Biological method and all already studied. Okay. These are the control measures. Okay. These two are the diseases on wheat. Only two diseases we are studying. Okay. One is the rust, second one is the smut. Kunkuma tegulu, kaatuga tegulu. In that, kunkuma tegulu is very, very important. Rust disease. Okay. Then we will go for the next is sorghum. Sorghum.
Yes, sir.
We completed rice and wheat. Okay. Then we will go for the sorghum, sugarcane, groundnut. Oh, three are there. Go. For the time now, already 9.
9.
Hmm. Okay, we have to complete.
Red groundnut.
Yeah. What are those? The sorghum diseases. What are those sorghum diseases? Two diseases we are going to discuss. Grain smut of sorghum, loose smut of sorghum. Both are smut. రెండు కాటుక తెగుళ్ళే. Grain smut, loose smut. Okay. Grain smut of
Yes,
sorghum
sir.
and loose smut of sorghum. Okay. Where we this over completed, this is completed. Yes. You see. The loose smut of sorghum. You see. This is caused by
Physiological Sorghu.
Physiolotica sorghum. Sorghum means sorghum. You know the sorghum, bajra. Sorghum sativum.
Sorghum vulgar.
Sorghum sativum is scientific name of the bajra. Okay. Jonnal. Jonnal antama.
Sorghum vulgar.
Sorghum vulgar.
Sorghum
Jonnal.
vulgar.
Jonna
Jonna.
rote. Sorghum. Physiolotica sorghum that causes smut disease. Okay. What it is? Smut disease of sorghum is caused by Physiolotica sorghum. That is the loose smut. Okay. Loose smut disease and the grain smut disease. We already discussed the loose smut disease in the wheat now. Same as like here also. You see, it attack first on leaf, then it enters into the phloem, then it goes to the pipe, It goes to the spike. That means pedicel. And then where it attacks to the all the pedicel, and it cover the black powdery structure upon the seed, so that it all the spike will be turned into black. Black color. కంకి మొత్తం ఎలా మారుతుంది ఇక్కడ చూడండి here. All the spike కంకి ఎలా మారింది బ్లాక్ కలర్ లోకి మారుతుంది. ఎల్లో కలర్ లో వైట్ కలర్ లో ఉంటాయి కదా మనకు బాజ్రా. Yes or not?
Yes.
That it
Yes,
turn
sir.
into blackish because it covered with the
Correct.
spores. Specialotheca spores
Spores.
fully covered with the seed, fully covered the seed. It get turned into blackish
Yes,
spike. Blackish spike. Okay, that is caused by Specialotheca sorghi. These are the symptoms. Okay, and then where it is over now. Yes. You see, sorghum, Specialotheca sorghi, loose smut. You see all the mycelial spores which are surrounded with the seed. You see the spike, all the spikes are turned into black. Okay.
Black.
And what are the control measures? Control measures, cultural, biological, and chemical. Cultural method.
Chemical.
Cultural method. What it is? Treatment of the seed before sowing. And healthy seed. We have to use the healthy seed. Disease-resistant varieties. And maintenance of the distance between the two crop or two plants. Pre-aeration, pre-sunlight. If otherwise attach all the plants, there is a chance to wide spread of the disease within the short span of the time. Okay, and then seed treatment I already told you, in the heat water or with the lime water or with the carbondism water, we can treat the seeds. Whatever the fungal spores, that all will die. Okay, and resistant varieties I already told you. And what are those? Next, chemicals, especially carbondism. And Titate Z78, carbondism, and Eucropast. Those are the chemicals which we use as fungicide to control the fungal diseases. Okay, this is the control measure. Okay, for the loose mud of sorghum. Okay, that is caused by Specialotheca sorghum.
Specialotheca sorghi.
Okay, then grain smut. That means that two are almost same, but small difference between these two is, here you can see all the spike will turn into black, but here in the loose smut, the spike, it is splitted. కంకి ఇట్లా ముద్దలాగా ఉండకుండా స్ప్లిట్ అవుతుంది, looser. That means some of the kernel this side, some of the kernel this side, 10 seeds this side, 10 seeds, 20 seeds this side. As like a witch's broom in the loose smut. But whereas in the grain smut, all the grains are attached in the only one spike. Only as a one bunch. But whereas in the loose smut, it's separate. Some of the bunches separate here this side. Some of the bunches bend this side. Some of the bunches bend straight. Some of the bunches right. Some of the bunches curved. Like that, spike కంకి లాగా ఉండదు. ఇటు కొంచెం అటు కొంచెం పోతాయి in the loose smut. But whereas in the grain smut, it is a bunch, only one bunch, but it all
It
covered
becomes
with
black.
the black pores.
Black. Mm.
Same Specialotheca sorghum.
Mm.
And they say
Hmm?
this is somewhat different. The splitted pipe kanki, virisku paina, that means splitted, separate అయినటువంటి kanki and bunched pipe. This is the difference. Remaining all are same. Okay. Remaining all the symptoms are same.
Okay, sir.
This
Okay,
is
sir.
the major difference between these two diseases.
Okay.
Okay. And then, the same biological control, chemical control, same as like that. Okay. Chemical control, carbonism, already told you. And biological and cultural is what? Seed treatment, wet, making the seed wet with the hot water and with the gypsum. Okay. And then next we will go with sorghum completed na?
Yes, sir.
So,
Yes, sir.
Yes, sir. Yes, sir.
sugarcane. One more disease, two
Next,
diseases,
groundnut, sir.
sugarcane,
Mm.
and one more is the groundnut. Sugarcane is red rot of sugarcane. Red rot of sugarcane.
Sugarcane
Yes,
leaves.
Colletotrichum falcatum.
Yes, Colletotrichum falcatum. Falcatum, sickle shape. కొడవలి ఆకారంలో ఉంటాయి. Posts are like a sickle shape. That's why we call it falcatum. Falcatum means sickle. కొడవలి తెలుసా? ఇప్పుడు కొడవలు ఎవరు వాడట్లేదు. అంత
Yes,
నైఫ్
sir.
Yes, sir.
నైఫ్
No, sir.
వాడుతున్నాం మనం కదా.
Sickle.
కోడళ్ళు ఎవరు కొడవలు వాడట్లేదు. నైస్ గా నైఫ్ వాడుతున్నారు. అవునా? ఎస్.
Sir,
కోడళ్ళు
sir, bookలో sugarcane leaves, sir.
షుగర్కేన్
లేదు సార్ గ్రౌండ్నట్ ఉంది.
వాట్ హ్యాపన్? షుగర్కేన్
డిసీజ్ లేదు సార్ బుక్లో.
ఉందమ్మా.
షుగర్కేన్ డిసీజ్ ఉంది, సిలబస్ లో ఉందమ్మా.
ఉంది 289 పేజ్.
289 పేజ్.
డిసీజ్ ఉందమ్మా. షుగర్కేన్ వన్ ఆఫ్ ద మేజర్ డిసీజ్. మీరు ఎక్కడైనా షుగర్కేన్ జ్యూస్ తాగుతున్నప్పుడు చూడండి, అక్కడక్కడ ఆ కేన్స్ ఉంటాయి కదా, కేన్స్ అక్కడక్కడ రెడ్ స్పాట్స్ కనిపిస్తూ ఉంటాయి. ఎప్పుడన్నా చూసారు? ఎప్పుడన్నా షుగర్కేన్ జ్యూస్ తాగారా?
Sir, yes, sir. We
Disease
observed in
observed.
cane also disease.
Disease
షుగర్కేన్ అక్కడక్కడ రెడ్ స్పాచెస్ కనిపిస్తూ ఉంటాయి కేన్ పైన.
Yes, tasteless.
ఇప్పుడు ఇట్లా షుగర్ కేన్ ఉంటది కదా, షుగర్ కేన్ ఈ తంపులు ఉంటాయి కదా మధ్యలో, ఇక్కడ మీకు అక్కడ రెడ్ కలర్ ప్యాచెస్ కనిపిస్తాయి.
Yes, sir.
Red rod of sugar cane. You see, if you peel out lower side, it all full occupied with the red. You see xylem and phloem fully covered with the fungi. It turn into red color. You see the leaves also turn into red at the middle wheel place get turn into
రెడ్
color. Okay, you see especially the nodal space, kanupu nodal space, it get turn into reddish color.
Yes.
By Colletotrichum falcatum. Colletotrichum falcatum. That this is the major disease in the sugarcane. Why commonly present? Almost 80% of the sugarcane which having the rod, red rod, which having the red rod disease. No, that is a common disease. It formed the acervulus. It formed the acervuli. Acervuli means the saucer shape. You see the saucer shape.
Sir, so much disturbance is there, sir.
Sir, your voice is low.
Disturbance is there. Not from my side. Another people are talking. The disturbance is not from my side.
Yes,
please-
sir, you know.
You please don't talk other while in the discussion in the main topics here.
Sir, your voice is low, sir.
My voice is low.
Yes, sir.
Ah,
No, sir.
No, sir, audible it is.
Audible, clear.
Clear, sir.
audible. My అరే నేను వాటర్ కూడా
Yes,
పెట్టుకోలేదు
sir,
నా
but
గొంతు
low, sir.
ఎండిపోతుంది
Yes,
నా
sir, but
గొంతు అంతా
low.
ఎండిపోతుంది నిన్న వాటర్ అన్న పెట్టుకున్నాను ఇలా వాటర్ కూడా పెట్టుకోలేదు పక్కన ఓకే రెడ్ రాట్ ఆఫ్ షుగర్ కేన్ that is caused by Coltotrichum falcatum that cause the red patches upon the leaves first and then it attack to the stem majorly it attack the xylem and phloem. The xylem fully loaded with the red color patches. The phloem, the green sugarcane green juice will turn into red color because all the phloem filled with the Coltotrichum fungi. It fungi characters, pathogen characters, it form the acervulus like fruiting body. Acervulus. You know, we have studied pycnidium, apothecium, perithecium, acervulus, ascostoma, fruiting bodies of fungi. We have studied sexual fructifications of the fungi. You see, open
Yes, sir.
not like saucer, just cup, teacup, cup-like structure, like cup-like structure. That we call as acervulus. That we call as
Acerulus.
acervulus.
Acervulus.
Say, you know the సేర్ అంటారు తెలుసా సేరు ఆఫ్ సేరు
తెలుసు సార్ తెలుసు
సేర్ ఆఫ్ సేరు కొలుస్తారు కదా బాటిల్ లాగా ఉంటది సేమ్ అట్లా ఉంటది ఇట్లా ఫ్రూటింగ్ బాడీ దట్స్ వై అసర్వులస్ దట్స్ వై వి కాల్ ఇట్ అసర్వులస్ సేర్ ఆఫ్ సేర్ బాటిల్ ఆఫ్ సేర్ కొలిసే దాని లాగా ఉంటది అసర్వులస్ ఓకే these acervulas which having the conidiophores. You see the conidiophores, bunch of the conidiophores. Conidiophores is the pedicel spike which form or cause for the germination of the conidia. That is conidiophore. Okay. At the tip of the conidiophores, which form the conidia. These conidia are half moon shape or sickle shape. Half moon or sickle shape. Sickle shape or half moon shape. Crescent shape antama, crescent or half moon. Crescent shape, sickle shape. Half moon shape or sickle shape. Okay, that's why we calling it falcata. Okay, sickle shape spores with a single nuclei that release again it attack on the next plant leaf. Okay, this is the pathogen characteristic. And what are the control measures? Same biological control. You know the biological control what?
Chemical.
We have to take the healthy cane. You know there is no seed for sugarcane. Sugarcane having the seed? Have you ever seen the seeds of sugarcane?
No, sir. No, sir.
Only
No.
With the notes.
Propagation.
Keep stem.
Only vegetative propagation. No sexual reproduction.
Vegetative propagation.
Yeah, only vegetative propagation. we cut the sugarcane into each nodal place, we cut into one. This will become the clone. We call it as clone. Clone. Vegetative seed pod, calling as clone. Vegetative seed pod, not the seed.
No.
If you see, it is a sugarcane. We cut in between the node. Suppose this is the one clone. This is another clone. This is another clone. With the node, kanupu ఉండేటట్టుగా కట్ చేస్తా.
Yes.
Each one will become the as a seed clone. While selecting that, we should take care that don't having any red spot. If the red spotted clones we soak in the soil, then again all the plant will be affected with the we have to take care while
Okay.
we have to remove this all up.
The disease.
Good. Okay, we have to remove this all. Yeah, next. What are those? Red dot of sugarcane I already discussed. Okay, you see, the process says there is a conidia in your, you see red spots. And what are the biological control? What are the chemical controls? You see here, disease control method, biological control.
Rhizobacteria.
We have to take the healthy clone, healthy nodal space. We have to, before soaking the sugarcane clone, we have to spray the ash. Boodigantama? Ash, katali boodida. Ash or gypsum. We have to spray the gypsum or ash in the soil so that we can control. Okay. And then next, what is the cultural? Cultural.
Yes.
What are the cultural practices? Of the clone treatment, cleaning of the whatever the clones with the gypsum or lime. Okay. And then chemical treatment, fungicide, spraying of the fungicide after crop. Before tilling, అంటే కంకిలాగా వేయక ముందు ముదరక ముందుకే at the early stage only we have to spray the fungicide so that we can control which one we control.
Yes.
We control your voice. We control your side top.
Disease.
Fungal disease. Okay, prevention is better than cure. So before tilling, at the early stage only we have to spray the fungicide so that we can control the disease. Okay. And then next, last, most important is Tikka disease of groundnut.
Tikka disease in groundnut.
Tikka disease, not Tikka.
Tikka.
Leaf spot of groundnut.
Yes.
Also called as
Leaf spot.
Leaf spot of groundnut. You can see here. The leaf spot. Leaf spot of groundnut. You see the spots, black spots. Black or brown color spots on the leaves. This also major disease, common disease in the groundnut. Major disease. Common disease, major disease in the groundnut. This is the major and common disease in the groundnut. That is leaf spot of Please follow.
Sir, please mute.
Please don't make a noise.
Don't disturb the class.
Okay. The only last we are going to the last disease it is. Okay. Don't worry. Don't bother about it. I'm taking your time much. 2 hours, 2 hours. Actually, I have to take only 1 hour class. They said only 1 hour.
Yes, sir.
But
Yes, sir.
yesterday 2 hours, today 2 hours. I'm spending my energy more than actual. I have to spend.
Thank you very much, sir.
Yes, sir.
Even in the class there also I spend 3 hours to
Thank you, sir. Thank you.
your classes. Actually, only 1 hour. Remaining all maybe take 1.5 hour. But I took
Yes.
3 hours to you.
Yes, sir. Yes.
Thank you very much, sir.
Thank you so much, sir.
Okay, now we are going to wind up the Zika disease of groundnut. Okay, the major disease, commonmost disease, and easy control disease. Okay, you know this disease is caused by Cercospora
Yes, sir.
arachidicula, Cercospora
Sonata.
personata,
Personata.
Cercospora personata, and Cercospora
Arachicola.
arachidicula. Early spot disease and late spot disease.
Yeah.
Early spot disease caused by Cercospora arachidicula, and late spot disease caused by Cercospora personata. Okay, early spot disease symptoms are uneven in shape, light brown in color, light
Exactly.
brown in color, light brown in color, and circular in shape, circular, round.
Round.
You see the brown color. Could you able to see this?
Yes, sir.
Clearly spots. You see the at the middle brown, it surrounds with the yellow. Yes,
Yes,
middle
sir.
at the brown, it surrounds with the white spot. It is there
Yes,
in the
yes, sir.
circular. You can see differentiation between these two spots. This is the round, circular, brown and yellow. Here only the black spots. Okay. These are the two differences. Early spot disease and late spot disease. Cercospora arachidicula and Cercospora personata.
Personata.
These two fungi causing the same disease, leaf spot disease, but symptoms are different. Okay. Okay. Here you can see the personata and arachidicola, you see same as like a saucer shape. You see the open saucer shape structure. See cup, open cup saucer shape structure. You see this is the saucer. Okay, saucer shape structure.
Okay, okay.
Bottom unna saucer. Okay. Then you see here, each one having the conidiophore. You see the conidiophore. If these all the conidiophores are even and bunched, it form the round circular spores. If these conidiospores are uneven, it goes this side, that side, bend, uneven size, uneven shape, go here and there, not in even. Then the spot also will be uneven shape, like this. Spot also will be uneven shape. If all the conidia are in one bunch in even space, all are in equal, that is early spot, round circular spot. Okay, these are the two differences between these two. Okay, if it widespread and here and there the branches conidiophore are here and there bends, it forms the uneven shape spot, like this, uneven shape spot. Okay, this is the commonmost disease. This we can control easily by using the gypsum, cultural method, common most method, cultural method. Gypsum, sunnam, gypsum and lime, of hydrated CaSO4 12H2O. Gypsum. That gypsum we have to use before soaking the seeds. విత్తనాలు నాటక ముందు, before soaking the seeds, we have to use the gypsum. And also we have to spray the gypsum in the soil, field, at the time of soaking. Then we can control this disease. Very easy to control, but very wide spread disease, very common disease in the groundnut. Very common disease. Okay, this is caused by two fungi, Cercospora personata and Cercospora arachidicola. You know arachidicola, Arachis hypoxia. Groundnut scientific name is groundnut scientific name is Arachis hypoxia. Arachis hypoxia. Hypoxia. Why they given the name hypoxia? మనకెందుకు, మనకు అవసరం లేదు, తిన్నామా అంతే. పైన పూసడి లోన కాసడి పుండరి సుమ అని ఒక్కడి సినిమాలో పాట ఉంది, ఆ పాట మనకెందుకులే. పైన పూసడి లోన కాసడి. యు నో గ్రౌండ్నట్ వేర్ ద ఫ్రూట్స్ జియో
అండర్గ్రౌండ్
జియో. జియో మీన్స్ ఎర్త్. జియో మీన్స్ ఎర్త్. హైపో ఎర్త్ లోపల ఉంటాయి.
హైపో అండర్.
Hypoxia. Why we have given the name? Seeds, fruits are inside the soil. Fruits are inside the soil. That's why hypoxia.
Okay.
వేరుశనగ కాయలు ఎక్కడ కాస్తాయి అంటే ఎక్కడ కాస్తాయి, మీరు కూడా అదే చెప్తారు ఇప్పుడు. చూడండి ఎక్కడ కాస్తాయి, ఆల్రెడీ మీకు చెప్పాను ఇంతకు ముందు క్లాస్ లో.
అండర్ గ్రౌండ్.
అండర్ గ్రౌండ్ లో భూమిలో కాస్తాయా? 100% అదే చెప్తారు అనుకున్నా. బాటనీ స్టూడెంట్స్ కాదు. బాటనీ స్టూడెంట్, you say the correct answer.
Symbiosis main is there na sir?
Root nodules, sir. Root nodules.
Yes.
Root nodules.
Root nodules, that is the another part. Root nodules that having the rhizobium that cause for the nitrification.
Nitrogen fixation.
Proteins, ammonia. You know, rhizobium bacteria which are present in the root. That root nodules that synthesize the ammonia.
Yes.
But what about the fruits? Fruits.
The end parts of fruits.
Fruits that are not the root crop. నేను పాట కూడా వాడినగా అమ్మా, పైన పూసడి లోన కాసడి పుండరి సుమ అని. ఫ్లవర్స్ పైన ఉన్నాక ఫ్లవర్స్ ఎక్కడ ఉంటాయమ్మా? రూట్స్ ఉంటాయా ఫ్లవర్స్?
పైన ఉంటుంది.
ఫ్లవర్స్ ఎక్కడ వస్తాయి? స్టెమ్కి వస్తాయా, రూట్కి వస్తాయా?
Branches.
ది ఫ్లవర్స్. ఎక్కడైనా ఫ్లవర్ దేనికి వస్తాయి? రూట్ రావు కదా ఫ్లవర్స్. పైకి కనిపిస్తే ఎల్లో కలర్ లో ఫ్లవర్స్ కనిపిస్తాయి, చూసారా? గ్రౌండ్నట్ ఫ్లవర్స్. మరి పైన ఫ్లవర్స్ స్టెమ్ కు ఉన్నాక ఫ్రూట్స్ రూట్ కొత్తయా?
స్టెమ్ మాడిఫైడ్ అవుతుంది సార్.
స్టెమ్ మోడిఫికేషన్. పైన పూసడి లోన కాసడి.
Stem modified food.
After fertilization, you know this, what it is? Pedicel, vruntam, todime. Avana? That enlarge, elongate, and the fruit, whenever the fruit forming, the fruit will be weight and బరువెక్కుతుందా? సో weight బరువుని పెట్టుకొని ఇట్లా పైకి నిటారుగా పైకి రాదు కదా. Whenever the weight increase, automatically it bends and it goes inside the soil.
Yes.
Goes inside the soil. After fertilization, whenever it getting mature, seed forming, what happens? Okay, then it goes inside. Okay, the stem crop, not the root crop. Don't say as a root crop. If you say root crop, you tear your degree memo. చింపి పడేసేయండి. రూట్ క్రాప్ అని చెప్పి పరువు తీయవాకండి. ఓకేనా? The stem crop, it flowers outside. After fertilization, the stem pedicel,
Root body goes to
the gynophore. The gynophore it enlarge so that it, whenever it enlarging because of the weight, it touch to the soil, and whenever it enlarging the gynophore, automatically it goes inside. You see, you pick up any one groundnut, whenever you see, you observe separately. You can see the roots are separate. Here are the roots. Here are the fruits.
Okay.
Here are the stem. So separate there. Don't get confused. Don't make others confused. Don't say false answer to anyone. Okay.
Okay, sir.
This is the common most disease of the groundnut. The leaf spot disease are also called as ticka disease.
Ticka diseases.
Ticka కాదు.
టిక్కా
disease.
Ticka disease.
No, ticka, TIKKA.
మళ్ళా లేకపోతే టిక్కలు వేస్తుంది. టిక్కా డిసీజ్ అంటే. Ticka disease. Ticka means, you know, the ticka means కాటుక.
Katka tegulu sir, meaning I mean
no, black color spots, but not the కాటుక తెగులు. Okay.
ఆకుమచ్చ
తెగులు, leaf spot.
ఆకు మడత తెగులు.
Leaf spot, not మడత, మచ్చ.
పూజ.
Not fold. Fold కాదు, fold అంటే మడత. Okay. And is caused by Cercospora personata, Cercospora arachidicula, and pathogen characters also we have studied. If the conidiospores are bunched and unique, that form the round circular part. If the conidiospores here and there it goes out, it forms the uneven shape part. Okay, these are the two different type of the spots. You know the control measures. Control measures, biological control, I already told you, using the gypsum, lime, add or seed treatment with the gypsum. If we do the seed treatment with the gypsum before soaking in the soil, we can control this disease. And also chemical control, Dithane Z78, and also gypsum, spraying of the gypsum. By spraying the gypsum also, and copper sulfate, gypsum spraying, we can control. We have to take the healthy seeds. Don't take the winkle folded అయినటువంటి ముక్క పోయినటువంటి seeds ని soak చేయకూడదు. You know, winkled seeds ఉంటుంటాయి కదా, ముడతలు పడి ఉంటాయి గ్రౌండ్ నట్ కొన్ని. You know, if you eat that winkle seed, you will got the botulism disease. Botulism. Headache వస్తుంది, stomach pain వస్తుంది, motions వస్తాయి. That disease called as botulism disease.
sir, botulism.
Botulism disease.
Clostridium botulinum fungi that are there in the groundnut seeds. And that means not all healthy groundnut seeds. Folded, wrinkle, small sized seeds. కొంతమంది తింటూ ఉంటారు కదా స్మాల్ సైజ్ వింకిల్ అయినటువంటి వాటిని. They get the stomach pain, headache, motions.
Vantams.
Vomitings also.
Vomitings.
Vomitings. Vomitings.
Okay.
Yes, all vomiting, motion, stomachache,
Stomach pain.
headache. Stomachache, stomach pain.
Okay.
That having the Clostridium, that fungi that cause this disease. Folded seeds, wrinkled seeds, folded seeds.
Sir, other name is botulism.
బోటులిజం మామూలుగా ఇప్పుడు చాలా హాస్టళ్లలో వస్తుంటది. హాస్టళ్లలో చాలా మందికి, ఎందుకంటే వాళ్ళకు చట్నీలు ఈ ముక్కిపోయిన పచ్చడితో చేస్తారు, ముక్కిపోయిన శనగలతో చేస్తారు.
ఓకే.
తిన్నారంటే ఏమంటే స్టమక్ ఏకు, వామిటింగ్, మోషన్స్ అవుతాయి. రౌండ్ పల్లీలు చేయాలంటే ఎక్కువ కాస్ట్ ఉంటుంది. వీళ్ళు థర్డ్ క్వాలిటీ పల్లీలు చేస్తారు. అన్ని ఉక్కిపోయినాయి, ముడతలు పడ్డాయి. That cause the stomachache. Okay, food poison. Okay, so we have to soak with the gypsum or seed treatment with the light heat water or copper sulfate, and you should do the crop rotation. Crop rotation is the most important. Even in the sugarcane also, every year should not stock the sugarcane. Next year how to crop other crop, even groundnut, even wheat also. So that whatever the remaining spores are there in the soil, if the diseased plant are there, even we pluck out the diseased plant, some of the spores will be remain in the soil. That will be cause the disease to the next crop. If you soak the same crop, again groundnut also, that cause the disease.
Sir, rotation crop.
It will cause the more damage. So that crop rotation is needed. If you
Crop rotation. Okay.
if you crop the groundnut, next we have to crop another crop, and then next season we can crop again.
Next another
Rabi Kharif is the both the time. కొంతమంది రబీలో ఖరీఫ్ లో
Other
both
different
times
type of crops.
groundnut వేస్తారు రబీలో ఖరీఫ్ లో. That cause disease. That cause widespread of the disease. Okay. These are the control measures: biological, chemical, gypsum, carbondism, gypsum, and copper sulfate or lime, sunnam. That is sunnam.
Fungicides.
Okay. That all are
Chemical methods.
Control measures. Okay, biological, cultural, and chemical. And the biological, one of the best method is disease resistant variety.
Yes, sir.
Okay, these all are the
Neem-based products also.
8 diseases we have studied. Okay, 8 diseases we have studied. First one is the Botany disease.
Botany disease on rice.
First one is the
Botany disease.
Bakra disease. First one is
Botany disease.
Botany. The second one is
Blast of rice.
Blast of rice. These two are historical, having the historical history for these two diseases.
Yes.
And important also.
Okay.
What is the gibberellin extraction from Botany disease plant and Bengal famine of the
Blast of rice.
Blast of rice. Bengal famine.
Okay. And then loose smut and grains of sorghum. Wheat rust.
Rust of
wheat rust and loose mud.
loose smut, rust and smut. Okay, rust
Yes,
disease,
sir.
kunkuma tegulu, and smut disease,
Katka
katka
tegulu.
tegulu. Rust disease is very, very important. Puccinia life cycle, macrocyclic life cycle, 5 type of the
Yes.
spore, compulsory question it is, and also blast of the rice also most important question,
Important.
Okay,
and
sir.
also ticka disease also most important question.
Yes.
From this
Yes.
Yes.
4 will,
Mm.
out of 4 may come, we have to write 2.
Okay.
Okay, these are the
Okay,
4
sir.
Ice blast.
most important diseases. Okay,
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 sir.
the remaining disease may come in the short answer as a life cycle of disease or disease control measure.
Sir, life cycle of disease, which one, sir?
No, if essay not come, it will come as a short answer. Suppose life cycle of
Loose master business.
or grain smart, life cycle of what it is, red rot of sugarcane, control measure of smart, control measure of rust,
Steam rust.
like that. So the question, 30 marks will cover from this lesson. Today, almost 2.5 hours, which cover 30 marks. Yesterday, 20 marks. Today, 30 marks. Okay?
Okay, sir.
Okay.
50
13,
marks.
sir. 13 marks, yes, sir.
30.
13. 30. Okay.
13 marks. 13 marks ఎక్కడ ఉన్నాయమ్మా?
No, sir. 10 marks
marks.
question 1, 5 essays.
5 marks.
So 30 plus 20, 50 marks
2
coverage,
major
sir.
diseases.
50 covered.
Short questions 10.
Sir, diseases plus life cycle ఏది చదవాలి సార్?
4 చెప్పు.
అరటిపండు తొక్క తీసి నోట్లో పెడతానమ్మా. అరటిపండు ఇట్లా తీయాలని చెప్పిన కానీ ఇక నోట్లో పెట్టమంటే ఇక నేనేం చేయని చెప్పు.
Sir, remaining 20 marks, sir.
గుర్రాన్ని నీళ్ళ దొరకపోగలుగుతాం కానీ మరీ నీళ్ళు దాపేయలేం.
No, sir, we have less time, but 4 papers read to sir.
Yes, sir.
4 పేపర్స్ కే అట్లా అంటే మరి 8 పేపర్స్ ఉంటే
Yes.
No, we are not spending so much of time to study, sir. That's why.
If you listen carefully, if you listen my lesson,
Yes.
you will be right thoroughly, no doubt at all.
Okay,
sir.
okay, sir.
Yes, sir. Yes,
Yes,
sir.
sir.
All
Yes,
the
sir.
Sir,
With the
please,
less time-
remaining 20
with the
marks
less time
also.
with the less time also, you can get a whole idea how to write.
Sir, 20 marks, sir. Remaining 20 marks from which topics?
Virus,
Bacteria is
bacteria,
there.
fungi. Remaining 50 marks from virus, bacteria, and fungi.
Okay.
Sir, just a small request, sir. Sir, some given to important questions, sir. Some I'm late joining, sir.
ఆల్రెడీ 210 అవుతుంది అయ్యా, నేను తినద్దా? మంచినీళ్ళు కూడా తాగలే, నిన్న మంచినీళ్ళు తాగిన.
Request, sir.
నేను కాలేజ్ నుంచి రాగానే ఇక్కడ కూర్చున్నా. 7:15 అయింది, అప్పుడు కూర్చున్నా.
అవును సార్.
ఆన్ చేసరికి 7:40 అయింది.
కొంచెం చెప్తే అయిపోతుంది.
Sir, which topics are important in virus, bacteria, and fungi, sir?
That I will keep in the group important questions.
Okay, sir.
Okay, sir. Thank you. Thank you very much.
Thank you so much, sir.
Thank you, sir.
Okay, then all the best for your exam.
Thank you very
Thank
much.
you, sir.
Thank you, sir. Thank you so much.
Some of them, half of them are already written the practical now. Remaining of we having the practical classes next now.
Yes.
Sir, tomorrow same class.
No.
Tomorrow no class.
Tomorrow, day after tomorrow.
C2,
C2, C3.
Monday, Tuesday again you have any classes.
Okay, sir.
Okay, we completed our first paper.
Yes, sir.
Thank
Yes, sir.
Thank you,
Good
sir.
topic.
Thank you so much.
Thank
Okay,
you,
thank
sir.
you so
Thank
much,
you,
sir.
sir. Thank you so much, sir.
Okay, then right.
Thank you, sir.
Sir,
Thank
thank
you,
you,
sir.
sir.
Right.
Yes, sir. Thank you. Good night, sir.
Good night. తినాలి. మీరు తిన్నదా తినుకుంటేనే మీరు వినొచ్చు. మీకు ఆ ఫెసిలిటీ ఉంది. తినుకుంటా వినొచ్చు. మీరు ఏం చేసుకుంటా వినొచ్చు. కానీ నేను అన్ని చేసుకుంటే చెప్పలేను కదా.
Yes, sir. అదే చేశాం సార్.
సార్ వచ్చిన డౌట్ సార్. సార్ ఇప్పుడు మనకు బెంగళూరు యూనివర్సిటీ నుంచి మనకు బుక్స్ ఇచ్చారు కదా, చదువుకుంటే సరిపోతుందా సార్?
సరిపోతుంది. వినఫ్. You listen the class, you can note down important points in that. You can underline the important points in the book. That are important points you have to write. That's enough. Need not to write a lengthy essay. Side heading should be.
Must.
All the side heading should be. Diagrams, wherever the diagrams needed, the diagrams.
Economic importance of bacteria important, sir.
Like yesterday told.
Economic importance of bacteria.
చిన్నగా ఏం చేస్తున్నావు?
Sir?
చిన్నగా ఏం చేస్తున్నావు?